Welcome to the Deep Dive. Today, we're tackling a really fundamental question in genetics. How do fruit flies stop their genomes, especially in the reproductive cells, from getting totally wrecked by these things called transposons?
Yeah, these jumping genes that
Um,
can cause chaos.
Exactly. And here's the really cool part we're diving into. Controlling them isn't just about how many copies are floating around. It turns out there's this specific inherited, almost like an adaptive immune system run by small RNAs, and it's coded right into the fly's DNA.
That's a great way to put it because, for a long time, the field was kind of split.
Right.
You had folks saying resistance just sort of builds up slowly the more copies of an element you have. But then there were these other findings about specific spots on the chromosomes, these discrete loci.
Like the famous flamenco region.
Exactly like flamenco. These acted like master switches for resistance. And the two ideas just didn't seem to fit together.
So there was this big gap. We knew certain proteins were crucial, the Piwi-family, things like Piwi itself, Aubergine, AGO3. They were essential for keeping the germline safe, and small RNAs were involved somehow. But the actual mechanism, the how, was really fuzzy.
So to get a handle on it, the researchers basically asked, what exactly is each of these Piwi proteins doing? What RNAs are they carrying?
How they figure that out?
Well, they used antibodies to, fish out the protein RNA complexes directly from fly ovaries. Then they sequenced just the small RNAs, the ones in that 23 to 29 nucleotide size range.
Okay, so they looked inside the machinery. What did they find?
A really clear division of labor. It was quite striking. Piwi and Aubergine, they overwhelmingly grabbed onto antisense piRNAs.
So the mirror image copies, the ones that can block the transposon message.
Precisely. About 76% for Piwi, 83% for Aubergine, and these antisense piRNAs often had a specific chemical tag at the beginning, a uridine or U.
Okay. But what about AGO3?
That was the flip side. AGO3 strongly preferred sense piRNAs, about 75% of its cargo. These have the same sequence as the transposon message itself, and, importantly, they didn't have that U bias at the start.
Huh. So different proteins are carrying fundamentally different types of instructions, antisense versus sense.
Exactly. That
Yeah.
asymmetry was the big clue.
Yeah.
It strongly suggested they weren't just working in isolation, but maybe interacting, maybe in some kind of cycle.
Okay, so the different players have different tools.
Yes.
But you mentioned this immune system idea, this genetic memory. Where does that come in? Are these piRNAs just made randomly, or are they coming from those special loci like flamenco?
Right. That's the next piece. They mapped where all the piRNAs that matched the genome perfectly actually came from.
Yeah.
Get this. Over 80% came from just about 142 specific regions, these discrete clusters, mostly tucked away in the tightly packed parts of the chromosome, the heterochromatin. And these clusters, they were stuffed full of fragments of old, broken transposons, like a graveyard. And crucially, yes, the classic flamenco locus was confirmed to be one of these major piRNA-producing clusters.
Wow. So flamenco wasn't just a statistical fluke. It's literally a source.
They proved it. They used flies where the flamenco region was disrupted and saw that the levels of one specific transposon gypsy just shot up like 20 times higher, and the piRNAs made from flamenco dropped way down.
So these master loci are actual literal memory banks encoded in the DNA, churning out these small RNAs to fight active transposons elsewhere.
That's the idea. They provide the blueprint for the defense.
Okay, I think I'm following. We've got the specialized proteins, PYO with antisense, AGO3 with sense, and we've got the memory banks, the clusters like flamenco, providing the raw material. But how do they talk to each other? What links the antisense side to the sense side to actually amplify the response when a transposon attacks? There must be a connection, right?
That is the core question this paper answered. And the answer turned out to be incredibly elegant, hidden in the relationship between the piRNAs themselves.
How so?
They looked at pairs of piRNAs that were complementary, one sense, one antisense that could theoretically bind to each other, and they measured the distance between their starting points, their 5' ends.
Then what did they find?
A huge spike at one specific distance. The most common separation between the start of a sense piRNA and the start of its antisense partner was exactly 10 nucleotides.
10. That's specific. Wow.
Yes, 10 nucleotides. And there was another clue. The AGO3-bound sense piRNAs very often had a specific base, an adenosine or A, sitting right there at position 10.
What does that 10 nucleotide offset mean?
It's the molecular footprint of an Argonaut protein slicing an RNA molecule. It was the smoking gun for what they call the Ping-Pong cycle.
Ping-pong. Okay, explain that.
Okay, think of it like this. An antisense piRNA loaded into Piwi or Aub finds an active transposon message, which is sense, and cuts it. That cut happens in a way that the new 5' end it creates is exactly where a new sense piRNA needs to start. And this new sense fragment gets loaded into AGO3.
Ah, so the cut product becomes the template.
It becomes the start of the new piRNA, yeah.
Yeah.
Now, this AGO3, loaded with its new sense piRNA, finds a precursor transcript coming off one of those memory bank clusters, which is antisense.
Like from flamenco.
Exactly. And AGO3 cuts that antisense transcript, and guess what? That cut generates the starting point for a new antisense piRNA, which can then be loaded back into Piwi or AB.
And round we go again. Ping-Pong.
Ping-Pong. Each cut generates the substrate for the next protein in the cycle. It's a self-reinforcing loop that massively amplifies the production of piRNAs specifically targeted against active transposons.
That's brilliant. So it elegantly connects the sense and antisense worlds and explains how the system ramps up.
And it unified the field. It showed how the master loci, flamenco, provide the initial templates, the memory, and how the ping-pong cycle explains the amplification, which connects back to why having some copies of transposon sequences around, in the clusters or active, is needed to fuel the response. It links both old models.
So the clusters are the memory archive, and Ping-Pong is like the rapid response team that copies the right defense blueprint when needed.
You got it. And it also neatly explains maternal inheritance, how moms can pass down resistance. They basically preload the egg cells with these piRNA complexes.
Giving the offspring instant immunity.
Exactly. It's a preloaded defense system.
Amazing. Okay, so let's recap the big takeaways from this deep dive. First, transposon defense isn't random. It's guided by these master control centers in the genome, these piRNA clusters acting like genetic memory.
Right. Second, there's a clear division of labor. Piwi and Aubergine proteins handle the antisense piRNAs, the blockers, while AGO3 handles the sense piRNAs.
And third, these proteins and RNAs work together in that elegant 10-nucleotide offset ping-pong cycle, which allows the system to massively and specifically amplify the silencing response against active threats.
Those are the key points.
Yeah.
It really laid the foundation for understanding this whole pathway.
It's a fascinating system, but here's a final thought to leave you with. While this paper beautifully explained the amplification loop, the ping-pong part, the initial step, how the very first piRNAs get processed from those long transcripts made by the clusters before ping-pong kicks in. Well, that process, known as primary piRNA biogenesis, is actually still pretty mysterious even today. Something to think about.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.